Um, so it gives me a great pleasure to introduce Russ today. I hope you've all had as much fun chatting with Russ as I did. And he comes to us from the, well, it comes to us electronically from the University of Santa Cruz. He is where he's an assistant professor. And he's going to tell us today a bit about his research. By way of background, he got his degree at Harvard, and he did his undergraduate degree in at UC Davis, and has basically been an emerging ecology evolution professor for a number of years. He has a stunning publication list in some of the most prominent journals, including Genetics, PNAS, Nature, Science, Science Advances, and has a number of awards, including the prestigious Sloan Fellowship. So it's going to be a great pleasure to hear him tell us today about his work. I'm sure you have discovered, as I have, that he uses the latest technologies and some model organisms to advance our understandings of some of the most fundamental questions in evolution biology. And also, he's very expansive in his thinking. He doesn't just think about the organisms he's working on. He's thinking about fungi, he's thinking about microbes, he's thinking about a wide array of animals, because his questions are very broad, which fits really well into the way we do things here at E3B. So thanks again for coming. Thanks for all of you for participating in these somewhat trying times. And I'm sure we look forward to hearing what Russ has to share with us today. Thanks, Russ.
Cool. Thank you so much for introducing me. And of course, thank you all for meeting with me and signing back in now to hear my talk. I'll just get straight to it if that's all right with everyone, and we'll go from there. But as you said, my talk is about how the genome got its shape, and I'll explain exactly what I mean in just a second as we go. If I can translate, there we go. Okay, so little bit of background. My field, broadly defined as evolutionary genetics, has a really long history of theoretical study. And back as early as the early 1920s, these two gentlemen, and of course many, many other people, but most prominently Wright and Fisher, had already really laid out a lot of the central questions that the field has still been working on to this day. So many of these questions have sort of gone unanswered, or in many cases we discovered the answer was a lot deeper than we thought it was, and we had to keep going. And so these are questions like, how does natural selection shape populations, and what are the molecular bases of natural selection? And as with many kind of early emerging fields, there was a lot of really big questions and remarkably little data. It's hard to directly address most of these. Remember that, back when they started talking about this, we didn't even know the structure of DNA. And so there was clearly a lot of basic biology we needed to get together to be able to go after these things. And there, of course, have been many, many important technological advances and revolutions since then, but the most recent, the one that's sort of driving my work forward, and I think a lot of other people's, is the genomics era, in which we're sort of firmly in the middle of now. And shown here are just two of the main drivers of this. It's an Illumina HiSeq and an Oxford Nanopore MinION, which is this tiny little USB device here. And one of the big outcomes of this has been what I'm calling a golden age for demographic inference. So this is a tree, sort of the evolutionary history of modern human populations and then ancient hominid groups that are related to them at some level. And the thing that I want to point out to you is just how incredibly, extraordinarily precise these approaches can be. So you've got cases on here, for example, this is gene flow between Altai and Denisovan Neanderthals of less than 0.5%. And these are actually two groups we've never sampled living examples of. They're known from bones from caves that have sat around for thousands of years. So it's pretty incredible how deeply and how precisely they can be characterized. And this of course, summarizes the work of David Reich and many, many other people. This isn't just one study, but it's still a pretty impressive feat that we have such a detailed map of human ancestry in recent times. And so in contrast, my research is trying to ask a slightly different question, which is how does natural selection shape the genome? So And I think you can probably guess from the image I'm choosing to represent this, that this is a little bit less worked out, right? We're not as clear on exactly how natural selection can play a role in shaping both the genome structure itself and genetic variation across the genome. And I also want to point out that when I say the word shape, I mean that quite literally. So a lot of my work is really focused on understanding genome structure and the way that genome structure can evolve over time and sort of the pressures that drive that. Okay. And to do this, my group uses a pretty broad range of tools. So we have long used sort of statistical computational modeling approaches on the right, and increasingly genome sequencing methods. So like I said, that's a MinION, but of course, lots and lots of different approaches. And then to some degree, molecular biology techniques to figure out how to sequence these things and how to do neat experiments. So kind of a combination of different things to get after this bigger question of what is natural selection doing to the genome. And with that, the talk is going to be structured into 3 sort of chapters, if you will. The first is about the gene expression impacts of chromosomal inversions. The second is called the modern origin of introns. And finally, we'll deal with our recent work on tRNA mutation rates, and calling it sort of cryptically the cost of transcription. And don't worry too much if these don't make sense yet. As I get to each chapter, I'll spend a minute to introduce it and what exactly I'm talking about, so it'll become clearer as we go. And with that, we'll get started on the first one here, the gene expression impacts of chromosomal inversions. So I like to start with a definition. What is an inversion? So what I'm drawing here are 2 chromosomes. You can see the centromeres in blue and the line coming off the chromosome arm. And an inversion is that red region where you can see the second chromosome has an inversion relative to the first. So the first reads ABCD, and the second is reversed in that region, so instead reads ACBD. It's reversed in the red chunk. And one thing that we have all agreed on, we think is the big effect of inversions, is that they suppress recombination. So if you had this individual and they were heterozygous for an inversion, so that, again, that middle region is reversed on one of the 2 chromosomes, during meiosis, if you had a crossover event, shown here within the inverted region, then the meiotic products that come out aren't very good. So you'll get 2 chromosomes, one of which is deleted for A, duplicated for D, and vice versa. But more concerningly, one of these chromosomes has 2 centromeres, one of these chromosomes has no centromeres. So that the idea here is that inversions play a big role in shaping recombination because they don't allow exchange or don't allow much exchange within the inverted region. And this has led myself and many, many other people to the idea that inversions can maintain supergenes, right? So the idea here is that if you have 2 genes, in this case, imagine they're little b and little c, and for whatever reason, little b and little c are a higher fitness when they're together, then that inversion suppresses recombination. It doesn't let them get onto different chromosomes. And so it will always be associated with these high fitness alleles, little c and little b. And conversely, if big B and big C are good together, this can be very beneficial for the population, and these inversions can get to high frequencies because of the way they restructure recombination across the genome. And there are a number of really beautiful examples. I like to show this image because I don't normally get to show pretty organisms in my talk. And so some really beautiful work from Duran et al. Back in 2011 took on this question in a big way. And what they're studying here is a family of butterflies, so the Millenniae. Those are 7 different species across the top there. There's 7 different color morphs in this family. And you can see that across the bottom are 7 different color morphs, all within a single species. Remember, each of these is a different species. These are all the same species. It's Heliconius pneumata. But you can see that each of these is mimicking the color morph above, and vice versa. And even more interestingly, so this is almost certainly many genes contributing to these phenotypes. But interestingly, in nature, you never see intermediate morphs. You only ever see this type of wing, never some sort of recombinant hybrid of the 2. And so the question is why? How do those stay distinct over time? And these folks actually answered it. And what they showed is that, at least for 3 of them, and probably for the others as well, that in the region that encodes these coloration patterns in the genome, there are these large-scale chromosomal inversions that are reversing large chunks of the genome relative to the other. And each of these contains some portion of coloration loci that distinguish those chromosomes. And so that's how they can maintain this complex mimicry pattern across a species range despite mating completely freely or nearly freely between these morphs. So it's a really beautiful example of what an inversion can do to structure complex phenotypes in nature. Okay, but what's happening at the molecular level? This has been much harder to address for many reasons. And what are the genes that are selected for? And finally, can we use differential expression to identify them? So that last one's a little bit more specific. It's kind of the approach I want to go with here, which is to use differential expression profiling to start to think about what genes are expressed at different levels between chromosomal arrangements and what that tells me about them. Okay, but there's a problem with that idea. And the problem is that there's actually two ways that inversions can impact gene expression. The first is the genome structure itself, which sort of has two subversions within it. In one case, you can rearrange large regulatory domains. So if we imagine the genome is structured such that all the genes in this blue region are co-regulated and they're all sort of freely touching in space. And the same is true in this green domain. When I mix them, because my inversion breakpoints are here and here, the expression patterns within the domain can be affected. Right, so based on chromatin state or other local epigenetic effects. The second is a little bit clearer. It's just direct breakpoint effects. So for example, an inversion breakpoint can simply break a gene, in which case you will certainly modify its expression, or it can just move local regulatory elements around such that perhaps the promoter of the blue gene lives here, but it now lives all the way over here. Okay, the second way that inversions can impact gene expression, and this is the way that I'd like it to be so I can study this, is that it could be that the mutations within the inversion affect gene expression. Right? So this is what we're trying to get at. Okay, but there's a problem there as well, and that is that natural inversions conflate genome structure with those linked alleles. Right? So when an inversion arises in a natural population, there will be thousands of unique or nearly unique mutations associated with it. And if it suppresses recombination, those alleles will stay associated with the inversion over time. And so it can be very hard to tell if the genes driving differential expression are associated with the inversion because of the history of the arrangement or because of natural selection. And so that was our major jumping off point. Our big idea then was to make some synthetic inversions, right? So what we want is a type of control, basically. And before I get into how we went about doing this, I'll just thank the people that actually did it. These are Victoria Serrano and Iskander Saeed. And what they did was they used an old genetic approach that's been engineered in Drosophila back in the early 1980s by a guy named Kent Golick. And so what he did here was there was a system that had been discovered in yeast called the FRT-FLP recombinase system. And the way this works is you have these sequences here and here on a chromosome. And they're called FRT sequences, which stands for flip-base recognition target sequences. And so what you can do is, if you construct this chromosome using standard genetic crosses to put these two FRT sites on a chromosome, if you then express flip recombinase, it drives ectopic recombination between those two sites. And so what that will do is it will generate a new chromosome. You can see that the red and blue have swapped, the A and B have swapped. So we've reversed the section in the middle between these two FRT sites, but it's genetically identical to its progenitor. It has the same big A and big B alleles. Literally the only thing we've changed here is the order of the genome. So you can think of this as a kind of control that we're going to use to distinguish the effects of linked alleles from the effects of genome structure. The idea being that natural inversions have both these structural effects and the effects of linked alleles, and these synthetic inversions really should have only structural effects. This will be the only method slide for this talk. Briefly, we had two natural inversions that we're going to look at. Each of them is relatively high frequencies in natural populations. And then there are two synthetic inversions that were designed to, as closely as possible, mimic the structure of the natural inversion. So their breakpoints are very, very similar, though not quite identical. And then for each of these, we made a bunch of replicates. So we'll be comparing standard homozygotes with inversion heterozygotes and inversion homozygotes. So we'll be looking at all three classes at once, and we're going to use the EdgeR statistical package to see what's differentially expressed. Okay, and again, to thank the people that did this work, Iskander Saeed and Ashley Byrne. Okay, and so you can kind of see the answer already, but I'll explain what this figure means really quickly. Each of these major columns here is one of the major recombining chromosome arms in the Drosophila genome. So there's 5 of them, 2L, 2R, 3L, 3R, and X. And each row is the set of differentially expressed genes associated with a given inversion, where IN3R SK stands for the synthetic mimic of natural inversion IN3RK right below it. And the same is true for IN2LST, the synthetic mimic of natural inversion T below it. And each point corresponds to differentially expressed genes along the genome. And so you can kind of see the answer to the big question already, which is that the synthetic inversions don't have a large impact on gene expression. There's a handful of genes across them, but we don't see a huge effect, and we certainly don't see breakpoint adjacent effects, which I'll talk about in a second. But in contrast, the natural inversions have fairly dramatic effects. Each has differential expression effects on the order of 100 genes across the genome. So about 1% of genes, or a little bit less. Okay. If we then zoom in and we look at the breakpoints, which I'm showing as these dashed lines, you'll see that, of course, there can be no enrichment because there are no differentially expressed genes for the synthetic inversions. In the natural inversions, there are genes that are differentially expressed around the breakpoints, but there's no enrichment for differential expression in those regions. There's just a couple genes here and there. But there is something really interesting and really consistent with this idea that linked alleles that are associated with the inversion might experience selection. So the thing we see first is that there is strong enrichment for differential expression on the same chromosome arm as each of the natural inversions. So you're much more likely to be a differentially expressed gene if it occurs on the same chromosome arm as the inversion itself. But interestingly, we also see that transacting effects, these would be genes across the rest of the genome that are not linked to the inversion, also are quite abundant as well. And if you do some sort of back of the envelope algebra for this, you'd say something in the range of 80% of differential expression effects that we see are due to transacting effects. Okay. And all of this is consistent with the idea that natural selection and not genome structure is driving these differential expression differences between arrangements, but it doesn't quite prove it. It could just be that there are natural alleles associated with the inversion. And so to demonstrate this, we're going to rely a little bit on population genetic theory. So the idea is that there is, if you had an arrangement, and so now let's imagine we're comparing genetic differentiation between, an inverted and standard chromosome in a population, where again, the breakpoints are these dashed vertical lines. What you'll see is that genetic differentiation is at its highest right around those breakpoint regions. And the reason for that is because recombination is the most strongly suppressed right at the breakpoint because there is no pairing. The chromosomes can't find each other during meiosis. In contrast, as you go away from the breakpoints, you'll get occasional recombination events that do shuffle the arrangement such that the later genetic differentiation going outside of the inversion and toward the center of the inversion decreases down to 0 at some point. But in contrast, if we had natural selection, so we have a gene, and this inversion is selected because it maintains this gene within the arrangement by linkage. If that happens, when there is another recombination event that occurs, those recombinants are selected against. And so the regions that are linked to that selected allele also are genetically differentiated between the arrangements as well. And you see it looks a lot like a breakpoint. It should kind of stick out. So if we then look, and now I'm only looking at the natural inversions, if we then look empirically at the data, the level of genetic differentiation across the genome, and again, each column corresponds to the major recombining chromosome arms, and in this case, the 2 rows are 2LT and 3RK. If I'm showing you genetic differentiation, you see that on the chromosome arm that has the inversion breakpoints, it's of course much higher than across the rest of the genome, which we would expect. The same is true for 3RK. But the interesting and maybe more important thing is that there's a very strong correlation between differential expression and genetic differentiation along these arms. So you see a lot of these outlier genes are at the tops of little peaks of genetic differentiation, which is exactly what we would expect if natural selection maintains these relationships. And in contrast, we see nothing, or very little effect at least, along the rest of the chromosome arms in this genome. Okay. So finally, I'll just say that we took these genes and we said, well, what do they do? All the differentially expressed genes for each arrangement, and we used a gene ontology analysis to look at them. What you'll see is that you get a bunch of different geoterms. I've just pulled out the subset here that I think are pretty interesting and have kind of informed my thinking about this going forward. You see that they're related to innate immunity and defense against gram-positive and negative bacteria in both of these inversions. And importantly, these aren't the same genes. They have 2 different sets of genes that they're both that both appear to be differentially expressed and associated with the inversion. So there's a really appealing hypothesis to go after here that these inversions may be involved in the species' immune response to bacterial infection. That, of course, is not shown at this time. This is just suggestive, but we have some follow-up work we're doing with collaborators that have thought a lot more about this sort of thing than I have. And I'm really excited to see where that ends up in a little bit. Okay, so to conclude that section, for these inversions, genome structure is not the major contributor to expression variation, but mutations that are linked to the inversions do cause differential expression at hundreds of genes, and this is both in the inversion itself and across the rest of the genome, so both cis and trans. The correlation of differential expression and genetic differentiation strongly suggests that natural selection is the force driving this, and the enrichment of differentially expressed genes for immune responses is a really tantalizing phenotype that we're excited to follow up on going forward, and I hope to show more results on soon. Okay. And with that, I'm going to transition quickly to the second part of the talk. This is actually pretty new work for the lab. It's something we've just got into recently, and it's actually completely unpublished. So, the figures are going to be a little less polished, bear with me, but I really like to always present something, unpublished and new and interesting. So hope you enjoy it. Modern origins of introns. Okay, and just before I even do this section, I'll thank the people that really drove this work. This is Landon Gazashti, a really talented student in the lab, and our collaborator, Scott Roy, on the right. Okay. So just a quick reminder of the molecular biology. Introns are the sequences that get cut out of the mRNA. So after your genome goes along and transcribes mRNA from the double-strand DNA, it'll have these exons, those code for your proteins, and it also has this intron here. And what's going to happen is it's going to get cut out by the spliceosomal complex, which is a protein RNA complex that comes along and cuts at the boundaries, pastes the exons back together, and makes your processed mRNA molecule. So introns get removed. But intron splicing and the way we do our splicing is actually super biologically important. There are lots and lots of cool examples, and I think maybe the best understood and one of the more interesting is this case in Drosophila, where differential splicing is basically the basis of sex determination in Drosophila. If you get this splicing event that joins exon 3 and 5 and skips exon 4, the individual develops as a male. It sets off a splicing cascade that has all sorts of expression effects, and you get a male. In contrast, if you get this splicing event where 3 and 4 are joined, you actually end up getting a female instead. So this gene is called double sex for obvious reasons, and it has this really, really cool and really important phenotype associated with it. So splicing matters. But interestingly, as important as splicing is, we appreciate this today. When it was discovered back in the late 1970s, it was kind of a mystery. No one really predicted splicing, as far as I know. And a big part of the reason is that, why? Why do we need this? When we have a perfectly good RNA molecule, why do we need to insert this stuff just to cut it out later? It makes, kind of no sense. And so this prompted a whole series of ideas, first identified as genes in pieces, and then why genes in pieces and why genes in pieces revisited. And actually, if you Google that phrase, there's like 10 or 15 papers that all have basically the same title, which I find kind of interesting. But importantly, all these theories, or most of the theories that tried to explain the origins and importance of introns, were really focusing on potential adaptive explanations without really considering where they would have come from originally and trying to think a little bit more about their proximal origins rather than downstream effects. And I think that's really the way we want to answer this question. We want to know, where did the introns come from originally? And of course, that question is also quite hard. Introns have been around for hundreds of millions of years. They're in all eukaryotic, or almost all eukaryotic genomes. So I'm not going to take on exactly that question. I'm going to say, where did introns come from recently? So for the subset that we can find, and we can figure out where they came from, what they do. Okay. So that's sort of the jumping off point to our work in thinking about the origins of introns, where they've come from and how they're evolving. The proximal molecular mechanism that I think makes a lot of sense and that we're working with is that transposable elements can generate intron gain. And the way this works is, if you have a transcript where these are my exons, the boxes, and these lines are introns, if this green line is a transposable intron, that is, it's a transposable element that can be cut out of the genome and cut itself back in somewhere else, if it then inserts in a different gene, so you'll see the structure of exons is different, it creates a new intron. And because it encodes its splice sites, this GT and AG, it actually splices correctly as an intron. And it isn't necessarily deleterious for the gene. It can just be removed by the spliceosome after this is transcribed from the genome. and you get nice, happy processed messenger RNA. And this has actually been recently shown in 2 species at least, and actually a handful of others that it's been hinted at, where you see extremely widespread intron gain. And it looks like what's basically happening is that you have a DNA transposable element that's transposed itself, thousands of times across the genome and has learned to encode its own splice site. So it's able to be cut back out after it cuts into the genome somewhere. Okay. And we're going to call these introner elements. And that's the question we wanted to ask, which is how common are introner elements in other genomes, and, and what determines their patterns of evolution? What fitness effects do they impose on the genome, on the organism? And what determines the distribution of introner elements across species? Okay. So as with many of my projects, this starts with a data mining exercise. So what we did was we went on GenBank and downloaded all of the annotated genomes I had at the time. I'll say that this number, 1,894, will get smaller through the talk as we remove things for quality control reasons. So don't worry, I am paying attention to that, but it will change a little bit, not to confuse you. Okay, and so what we do first is we download all those genomes and we extract the regions of those genomes that are annotated as introns. And we then do an all-by-all comparison. So we're using a program called BLAST, which basically searches for sequence similarity between each of these elements. So each pair is compared. And because it's a full pairwise comparison, you'll see that this basically encodes a type of graph where what I've done here is removed all the links between elements that aren't that genetically similar and left links between those that are fairly similar. And what you can do is pull out these sorts of clusters of a bunch of different sequences that are all way too similar to each other that we would expect by chance. And then if you make the tree out of these sequences, which we did later, you'll see that, yes, in fact, they encode a tree like you might expect for a bunch of sequences that are related by common descent. And even you've sort of got 2 clades here, light blue and green, where blue is nested within green. Okay. And then what we're going to do is we're going to go through here and we're going to make sure that when we find these cases of that we think are intron gain, that it really is. So if I compare this species gene structure to several outgroups, so to its closest relative and 2 outgroup species, what we should see is an event like this, where you see an intron only in this most recently studied species that isn't present at any position in any of these other species. That's pretty good evidence that we found a real mechanism driving intron gain. Okay, so what do we find? We looked through a bunch of species. Of them, you find a good amount of recent intron gains driven by intron elements. So about 2.5% of all species that we looked at fall into this class. They're not randomly distributed. It's actually highly variable across eukaryotes. So what this graph is showing you across the x-axis is a number of species in a given group. These aren't quite clades, they're grades in some cases, so group of species that have active introner elements. And then on the y-axis, you're seeing the number of species that do not have active introner elements, or that do have active introner elements. You can see that some groups are real outliers here. So the fungi tend to have both a lot of sequenced species, but also a good amount with active introners. If you look over here at the Cnidarians, they're a pretty interesting group in the sense that they actually have, 80% of the Cnidarians we looked at had active introner elements or recently active introner elements. So a really appealing group to look at going forward and something we're digging into more and more. And I'll return to this later. So I want you to think about this, is what's special about these species along the top here. Okay, if I'm now plotting them all on a tree, this tree only shows the species that we did find introner elements. I'm obviously not showing all, almost 2,000 species here. And now at the base of each branch, you're seeing a point where the width of that point corresponds to the number of introner elements in the genome. There's some pretty interesting variation across groups. So for example, just between ascomycetes and basidiomycetes, you see a pretty dramatic difference in how frequent introners are, where most of the ascomycetes have little points and therefore not many introner elements, and most of the basidiomycetes actually have a ton of points, so many introner elements. Okay, if we then look at the proportion that occur within genes, you'll see that this also varies quite a bit as well. So for example, the fungi, if this box plot represents the null model from permuting the positions across the genome, the red line represents the actual number observed within genes. You see that the fungi are super enriched for having introner elements within genes. In fact, there's a few that have 20, 30 introner elements that only insert into genes, like here and here. In contrast, the animals are almost exclusively enriched, with one exception, for being well outside of genes. suggesting potentially that these are actually quite deleterious in animal genomes for whatever reason. Okay. But we'd like to take a little bit more quantitative view of natural selection. In this case, what we're going to do is we're going to use RNA-seq data to quantify splicing efficiency. So for every one of the species we found introners in, we downloaded all the RNA-seq data we could, and we started looking at how well they splice. Where I'm basically asking, for good splicing, did you use the correct annotated splice sites at the 5' and 3' end of the intron element and make the RNA molecule we think you should? Or did this particular transcript select a different 5' element, for example, in this case, and make a truncated protein coding sequence down here? Okay, and there's some more predictions here. So we want to know how frequently bad splicing occurs in general. So in this case, what we're going to do is we're going to compare the introner introns to just canonical introns that do not appear to be recently gained in the genome, and look at the ratio of bad to good splicing. And finally, what we're also going to do is look at high expression versus low expression genes. The idea being that if you're an introner and you splice badly, a high expression gene is going to do a good job removing this. And so you'll lose that introner element before it goes to fixation in the population. In contrast, for a low expression gene, that shouldn't happen, or should happen less frequently, and you should accumulate introners in those genes. And that's exactly what we see. So here what I'm doing is I'm showing you all the species again, but I've now subdivided them into the set where their introner elements splice worse than canonical introns. And on the right are the set of where introner elements actually splice better than canonical introns. And again, this box plot represents the permutation, where if I just threw these things down at random, what would be the expression level of those genes? And then we normalize it so they all have a mean of 0. So they're all sitting at 0 for permutations. And what you can see is that for the subset that splice worse, many of the species, more than half of the species, are in much lower expression genes than you would expect by chance. These red lines down here at the bottom. But conversely, for the subset that splice better, that almost never happens. In fact, the vast majority of them don't show any significant differences from our permutations, with the exception of this one, which actually splices better and is in higher expression genes. And I don't know why, but I think it's quite interesting. Okay. So to answer our questions from the beginning, do introner elements drive the accumulation of introns? Yes, almost certainly. So if we were to subdivide these two groups and count up all the introns in their genomes, and we divide them into the set of species that have introner elements on the left and the subset that do not have introner elements on the right, what you'll find is that, yes, we have more introner elements within the group with active IEs and more introns total in the group with active IEs. And more importantly, this remains significant even after a phylogenetic correction using RevBase. So we see that there really is a very strong correlation across life. Okay, what are the factors that drive the phylogenetic distribution of introner elements? Remember, we started with this figure at the beginning, and a different way to ask that question is to ask what's special about these taxa in the red box here. So why do these taxa in particular seem to accumulate introners at a really high rate relative to the rest of taxa along the bottom here? And this, I'll just say, is conjecture completely at this point. It's not proven, but I think it's possibly an explanation. And the idea is that if a species has an accessible germline, this could facilitate introner gain via horizontal gene transfer. So if you have a species that for example, is single-celled, like many algae, then that germline is inherently accessible because their only nucleus is their germline, and it will get in at some rate. In contrast, if this introner tries to insert in this organism where only one of these cell lineages is destined to become the germline, it may be much harder to be stably inherited in the population and then much less likely to be transferred between species. And I'll just say before anyone asks that we have not identified a really clear-cut case where this has happened. So I'd like to see two species with almost identical introner sequences. But it's something we're looking for more and more. And we do see quite a few cases with excess shared motifs, sequence motifs, and other things that at least look like it might be relatively recent transfers. And if we want to test this model, we see that, yes, there's actually a fairly strong effect. About 5.5% of species with accessible germlines and about little less than 1% of species with inaccessible germlines are likely to have introner elements. So there's a pretty strong skew there. And this also is fairly significant after a phylogenetic correction. I will caution you, though, that there are a ton of other factors that differ between the taxa that have these and don't. And I cannot at this point really prove that it's something to do with the accessibility of the germline. But I think it's a really tantalizing hypothesis, and it's something that we're excited to keep working on going forward here. Okay, so to summarize this section before we move on, transposition is clearly driving intron gain. You know, IEs are abundant. They're in about 2.5% of all species that are annotated on NCBI. Negative selection shapes the distribution of IEs within genomes. So at least for the cases that we can find evidence of selection, it looks like selection is mostly trying to remove introners as they arise. So the evolution of introns might be deleterious, at least by this mechanism. And 3, horizontal transmission and germline accessibility might shape the distribution of IEs among species. And if IEs are the primary mechanism driving intron gain, then these are basically the factors that shape the distribution of introns across eukaryotes, which I think is a really interesting and potentially very important observation, but we got to prove it. Okay, and with that, we're going to go to the last section of my talk. This is a little bit cryptically titled, as I said. It's the cost of transcription, but I'll explain in just a second what that means. And it's our relatively new work studying the evolution of tRNA genes in humans and actually many other species as well. And to thank the person that did the work, this is my student, Brian Thornlow, who's doing just absolutely amazingly in his PhD, as you'll see in a second. tRNA genes are life's adapter molecule. I'm not a great artist, obviously, so bear with me and just try to follow this image. But if we imagine that this line along here with the poly A tail is a messenger RNA, and that messenger RNA is being translated by this ribosomal complex shown in blue, which is itself shepherding in these tRNAs and attaching their corresponding amino acids to the growing peptide chain. That's basically translation. So they're an adapter molecule. They go from RNA to protein. One thing we know for sure about tRNAs is that they are extremely highly transcribed. So something like 15% of total RNA in growing mammalian cell culture is actually tRNA transcripts. So when you consider that something like 82% is ribosomal RNA, you realize that actually only a tiny amount of the RNA that we study and we focus so much on, messenger RNA, is actually the RNA that's in the cell lineages, strangely enough. Okay, something else that we know about transcription. So tRNAs must be extremely highly transcribed. Transcription is itself a mutagenic process. And this has sort of 2 main ways that it works. In the first, what happens during transcription is that as your DNA duplex is being unwound, and you have this RNA molecule that's forming along the coding strand of the DNA here. Along the top, you have the non-coding strand of the DNA, and this is just sort of hanging out as single-stranded DNA freely in the nucleus. So it's not protected by protein complexes, and it's single-stranded, so it's pretty unstable. And during that time, it's exposed to potential mutagens. Similarly, you can have conflict between the replication and transcription machinery, where they either hit each other in head-to-head orientation, or one isn't moving fast enough and the other catches up. And these can induce torsional stress on the DNA, which also can induce mutations. You know, tRNAs may be very, very highly mutated, potentially by transcription-associated mutagenesis, but they're also super important. So they must be very, very highly constrained by natural selection. And I'm showing you here sort of the basic anatomy of a tRNA locus. They're polymerase III transcribed genes, which means that they have internal promoter regions. So your A and V box are super important sequences for transcribing the gene. But even more importantly, they have to fold in this really characteristic cloverleaf pattern, right, where you have these 4 stem regions. You also have many regions that have to do important functions. So for example, that's your anticodon, which actually binds the mRNA molecule. And of course, this section up here binds your amino acid as your peptide chain is being grown. So this sequence is super important, super highly constrained by natural selection. And just to show you that, if you induce even a single mutation to this thing in just one of these stem regions, you get a completely different predicted folding structure and presumably a completely non-functional tRNA. So natural selection really, really wants to maintain them the way they are. But what's really cool about tRNAs is that their flanking regions are also transcribed, but they're mostly neutral. So if you induce sequence mutations in these, it doesn't affect the tRNA's expression. And because they're removed in a sequence-independent manner after transcription, it doesn't seem to matter what is going on with these flanks at all. And so this is basically a population genesis dream, right? I have a gene in the middle. that I really want to know a lot about. I want to know how it works. I want to know how natural selection is working. And then I have these regions on the edges that experience very similar pressures. They are transcribed, but they don't experience any detectable selection. So there are neutral markers. Okay. So the first thing we're going to do then is we're going to look at rates of polymorphism. So what I'm looking for now are low frequency SNPs, so single nucleotide polymorphisms, in human populations. So we're looking across a pan-African data set of a few thousand African individuals. And what I'm plotting here is an aggregate analysis. So I've taken the approximately 400 tRNA genes in the genome, aligned them all to each other, and then I'm showing you an aggregate of the proportion of tRNA genes that have a low frequency SNP mutation at a given site in the genome. And what you can see is that the 5' and 3' flanking regions, which again are transcribed but are removed enzymatically, do have a super high rate of these SNPs. And the tRNA gene itself is also fairly high when you compare it to the genome-wide average, which is this dashed black line along the bottom here. So it looks like there's an excess of mutation. We see quite a lot more mutations within tRNA genes and at the flanking regions than we do in the genome-wide average. But if we then look at divergence, right? So divergence is shaped not only by the mutational processes, but also by natural selection, because to have a mutation go to fixation between species, it must be either not very deleterious or even good for you. And so now what we're doing is we're comparing humans and macaques. What you can see is that, again, the 5' and the 3' flanking regions, which we think are neutral, have a hugely elevated rate of divergence between the species. But the tRNAs themselves are actually much less divergent than the genome-wide average. So natural selection has done its job. It's removed those low-frequency mutations, and we don't see a lot of fixations between humans and monkeys. So all of this is consistent with the idea that transcription-associated mutagenesis is the process driving mutation, but it actually doesn't really prove it. And so to do that, what we did was we took tRNA genes and we subdivided them into two sets. It's the same kind of analysis again. We're again looking just at divergence, but on the top I'm showing you the tRNAs that are highly expressed. So they're in regions of active chromatin in basically every tissue. And on the bottom I'm showing you the tRNA genes that are not expressed, or at least we don't know of any evidence of expression in human genome at all. And what you can see is that the pattern is even more distinct if we do this, where the tRNA is actually much more strongly conserved within the highly expressed genes. And then the flanking regions are even more highly divergent, again, within the highly expressed genes. And across the non-expressed tRNAs, you see that the divergence is almost exactly the genome-wide average, more or less. We do see a slight excess of divergence in the 5' and 3' flanks, which I would suggest to you just means I haven't done a perfect job of subdividing the tRNAs. But regardless, this is very strongly consistent with the idea that transcription-associated mutagenesis in particular is driving these hugely elevated rates of mutation at tRNA genes. An obvious question to us was, well, is this something weird about humans and primates, or is this a conserved process of tRNAs themselves because of transcription-associated mutagenesis? And what you see, what we did to ask that was, we looked across 3 model organisms, so that's mouse, Drosophila, and Arabidopsis, and of course human at the top. And you see, at least qualitatively, very similar patterns of conservation, where the tRNA genes are very highly conserved. In fact, they're extremely highly conserved in Drosophila and Arabidopsis, but the flanking regions have an excess of divergence relative to the genome-wide average. So again, we're seeing this pattern across all these species, suggesting this really is a process of tRNAs themselves and not some unusual feature of humans. This last part of this section is a bit of an aside, but I think it's really cool work that we're pretty excited about, just because we get to now make functional predictions about the genome. The idea here is that, if this process is really dominating patterns of evolution at tRNAs, then we should actually be able to predict tRNA gene activity just using patterns of sequence conservation and anything else we can learn from the genome sequence itself without ever actually functionally measuring it. And so obviously in humans, that's not important really, because we already know which tRNA genes are on or off. But across the vast majority of organisms, so basically every other mammal except mouse, we have no idea which tRNAs are on or off. And if we could use this sort of information, we could make those kinds of functional predictions without ever directly measuring it through really, really painstaking and laborious experiments. And so what we did, if you've been listening, you probably could make a guess about whether or not this is a good predictor. You see that the super highly conserved tRNA genes here, in this case, sequence conservation goes up, sequence divergence goes down. What you see here is the super highly conserved tRNA genes are also the most highly expressed. So these 4 are active, they're very highly conserved, and they have these strong depressions in the flanking regions. You don't see this for the 2 inactive tRNAs, or much less effect. Okay. And I won't go into great detail about this. We've just been getting into some machine learning, and this was our first foray into it. But we're using a really simple random forest classifier algorithm to do this sort of thing. And I'm just showing here a set of features. These are both features that are extrinsic, so they're about the tRNA itself. Or sorry, they're about the region the tRNA is in, and then the subset that are intrinsic, so they're about the tRNA sequence itself. The features aren't that important, just to show you that we're using 10 total features to try to do this. And when we do this on humans, doing kind of standard machine learning training where we hold back 10% of the data and we try to predict those 10%, we see that we do pretty good. It's about 93% accurate on humans. But that kind of doesn't tell you very much because, of course this works on humans. We trained it on humans. So what we did then was we took a model we trained on human tRNAs and we fit it to mouse tRNA genes. What you can see is that the accuracy is about the same. It's about 93% accurate applied to mouse. So it really, really is a very strong predictor of tRNA gene expression, and we're able to do a pretty good job distinguishing active and inactive tRNAs. And if you want to read more about this, it's just come out recently in Genome Research. You can look it up on my Google Scholar. Okay. And so to return briefly to the first observation, well, there's some really key implications for human fitness. If you have a 10 times elevated mutation rate across tRNA genes, it implies that in aggregate, tRNAs contribute to about a 1% decrease in fitness due to mutations in tRNA genes. So this is sort of classic mutation load theory type stuff we're using, which is really amazing. So to summarize this section, so again, this suggests that tRNAs are really a fundamental driver of fitness variation in humans and probably just about every species because we see that same basic pattern across life. All right, so to summarize this section, transcription-associated mutagenesis drives mutation rates that are 7 to 10 times higher than the genome-wide average. It depends how you do your math. This pattern is consistent, and it's actually even predictive across species. And so far what we've done is applied this to 30 mammalian genomes that we were able to download and analyze. But in the future, we're hoping to apply this to a whole bunch of other things as well because mammals are one challenge, but if we can build a model that's even more generalized than that, we could predict tRNA expression levels across all of life, which would be even more interesting. And importantly, this implies that tRNAs have vastly outsized fitness effects relative to their length. So, tRNA gene is 0.0006% of the genome or something like that, but if they contribute to 1% fitness loss in the average human, that's big. Okay. And finally, this is kind of leading to the last section I'll do here about future directions. This implies that tRNA genes are a really great model for studying the effects of strong mutation and strong selection. So most population genetics assumes mutation is a very weak force and selection is a very weak force. Here, it must be that mutation and selection are quite strong. Okay. And so this is a section coming soonish, a little preview of things I'm thinking about with tRNAs. I'll just say that this is very much work in progress, but I think it's got a lot of room to run. I think it's a really important theory to take on, and tRNAs are really a cool model to use for it. Most evolutionary theory is concerned with the germline. So we imagine there's this blue cell lineage that dictates the fitness of the entire organism that develops, and it's passed on to its offspring. Germline and mutation alone doesn't really explain functional redundancy. So something I didn't mention earlier is that tRNAs are present in the genome at enormous copy numbers. So you have something like 20 of certain tRNAs, you have 20 copies across your genome. So there's quite a lot of them, and they appear to be functionally redundant. Okay, and selection for redundancy, when it's just the germline mutations, is actually quite weak. The reason is, if these two genes are exactly redundant, then having only one copy doesn't matter. This germline lineage is just as fit after it loses one as it would be with two. So the selection to maintain redundancy is about as strong as mutation to remove it, which tends to be a weak force for many things. And over time, what should happen is the genome will revert to single copies. You'll only have one of each functional element. So this is essentially the basis of Mike Lynch and other folks' arguments about how genome structure should evolve. But the genome has absolutely tons of tRNAs. So what's going on? And the answer, or I think a possible answer, is that somatic mutation has enormous fitness effects, and that's what's driving the evolution of redundancy. So that blue lineage doesn't exist on its own. Those germline lineages require this whole set of somatic lineages to be successfully passed on. And in large multicellular organisms like humans, there's a heck of a lot more somatic lineages than there are germline lineages. And mutations that occur early in development are inherited by the full set of downstream tissues. You can see it in quite a lot of tissue. Mutations that occur later might contribute to senescence. So they'll be in fewer cell lineages, but you get enough of them and eventually you start senescing. Importantly, this can all be mitigated by the presence of genetic redundancy. So if instead of having one, I have two elements, then that somatic cell lineage is just as fit as it was previously after it loses one of these functional copies. And for a gene that's super highly transcribed and experiencing very high mutation rates, you can imagine that's quite important. So I think this is one of the major factors driving tRNA redundancy and evolution across species. And for theory on this, you should check out some really cool work. Alan Orr thinks this might be the basis of diploidy in the first place, and Martin Novak's theory is really the basis for this work. And so the idea here is to put the theory to the test. And it's actually remarkably straightforward. It basically comes down to this, which is that if the somatic mutation rate is greater than the germline mutation rate, then genetic redundancy can evolve. And to do this, we're making some specialized, nearly error-free sequencing libraries to really directly and quantitatively measure the somatic and germline mutations. All of this to ask the sort of bigger question, does somatic mutation drive the evolution of genetic redundancy? And maybe more generally, to start to link developmental processes to genome structure evolution, which I think is a really important frontier. And with that, some quick acknowledgements. I'm at the end here. On the left here, all the people that really did the work, and the right, people in my lab and other labs that have really contributed to this, and of course my sources of funding. And thank you all for coming.
Thank you. So we have, we can open this up for questions now. We've been using a hand function, which if you show the list of participants, you'll see there's a hand that you can.
Okay. Yes, I found participants, and I can. There are hands on here I can click on somehow.
Okay, so Andres has his hand up, for example.
Okay, there we go.
Thanks for the very cool talk. I have two questions. One is on the, can you do a similar analysis for genes that are in high copy number, like histones versus tRNAs, and how do you incorporate the need for, one of the hypotheses for high copy number genes is that you need to produce lots of proteins rather than the somatic redundancy. How do you incorporate that into the?
Yeah, that's a great question. So the first part of that I'll say is absolutely it works. So if you want to know about histone specifically, that's the gene we looked at, and it's in the supplement of the paper. But basically, you see qualitatively very similar patterns as you do with the tRNAs, where the flanking regions are crazy divergent and the gene is pretty strongly conserved. I'll say the one trick though is that with a protein, it's much harder to know exactly where it starts and stops. And so it's trickier to do this analysis in high throughput automated ways, whereas tRNAs have this nice feature where I just know where they end, and it's pretty easy to see this sort of pattern. But absolutely one can do this with proteins as well. And it's something we've been working on, but it's harder. The second question, sorry, I'm not thinking.
No, I guess I only asked one of them. I was gonna wait to ask the second question, but I see Dustin has his hand up. So I can ask after Dustin, my other question.
Oh, I think you already, I think I'm remembering it now, actually.
Okay, there were two parts to the first question. Sure, go ahead.
Sorry, the second question, to remind everybody really quickly, was, how do we know that it isn't just that you need a ton of genes so you can make protein? You got to juice them really hard, basically.
Yeah.
This experiment's been done in yeast at least, and there is some work in mouse as well, although that's a little bit more equivocal, where if you knock out a tRNA that has another copy in the genome, nothing happened to the growth rate of those yeast. But if you knock out a tRNA that's unique in that genome, they die. So at least in that context and that specific assay, it looks like it's probably functional redundancy. With that said, I'll just say that the models we're building in to fit this include the possibility that you have, that you need more than one copy at a gene. And we're trying to think about how to tease those signals apart. And I think there are ways, but it's a little more technical than I want to do right now. But yeah. Cool. Dustin?
Okay, you done, Andres? So one of the things that's always fascinated me about inversions is that multiple genes seem to accumulate on the inversion. And one of the hypotheses is because of transposition from transposons and stuff. So I'm wondering, when you made your synthetic inversions and compared the natural inversions to the synthetic inversions, did they have the same genes, or was there an accumulation of other genes on the natural inversion that might have explained some of those results?
That's a great question. So I think in this case, it's probably unlikely. The two natural inversions I picked are inversions I studied pretty heavily in my PhD. And we've estimated their ages, using standard population genetics kinds of approaches, and they're pretty new. So they're pretty young inversions evolutionarily, although it's still possible that a number of genes have transposed over. It's unlikely that it's a lot of genes that have transposed over. And we've actually looked for secondary structural rearrangements by using discordant read pairs and other things, and we don't see much. But it is possible that some of those genes are doing that. I think probably not most, though. Yeah.
I have one more question.
Sure. Right.
In the inversions, the patterns of when you were looking at divergence around the inversions in the chromosomes, I think you see very high signal throughout the chromosome in divergence, not necessarily that much higher within the inversion. Can you talk more about it, what that means, or what it implies, if there's long range regulation or other patterns of recombination that are more long range than just within the inversion? Or...
Yep, that's a great question too. So the answer is we think it's mostly to do with the fact that inversions suppress recombination both inside and outside of the inversion. So I give the kind of simple explanation when I'm setting that up, but they're tricky is the answer to inversions. And there's some really cool work from Mohammed Noor, and then also population genetic work I've done in the past that shows that, yeah, it's pretty clear that recombination is suppressed, up to 4 or 5 megabases outside the inversion breakpoints as well. They do recombine, just at much lower rates, basically. And so I think that's probably a big part of why you see that excess differentiation going out and in the middle. Cool.
I don't see a hand up yet, but I'll ask a question.
Sure.
I'm a biodiversity guy, and I'm assuming you can hear me since you're nodding. I worry that my microphone is
hearing you.
By the way, really, really fascinating talk. Thanks for that. And, I'm thinking about biodiversity. I love the idea that one can actually get a much higher resolution handle on some of the mechanisms that actually drive diversity. And I think your work is actually beginning to shed some light on that issue, on those many, many issues. But, when thinking about it, and I think of it at a very, very coarse level, because I'm mostly focused on, phenotypes and functional diversity and so forth. It's often for the really interesting parts of diversity are changes in developmental genes. So I was thinking about, things like Hox genes or MADS box genes in plants, right? So do you think the same sort of things you're observing with something as fundamental as the tRNA, genes is going to wind up playing out the same, where you have, the amazing thing about the Hox genes is you have that very same sequence found across, whether it's invertebrates or vertebrates, and yet the changes there can be dramatic, getting you things like bithorax and
yeah.
So what's your thoughts? It's just a very broad question. What's your thoughts about whether or not this is going to, these sorts of things you're finding out are going to play out for some of the gene functions that actually control major shifts in diversity, biodiversity?
Right. Well, tRNAs kind of live in a slightly distinct space from proteins, right? So proteins have this thing where they interact with, Hox interacts with a lot, but most proteins interact with just a handful of other genes, right? Directly at least. Obviously, they make an organism, so they must interact at some level, but the direct protein-protein interaction, whereas tRNAs actually simultaneously interact with everything. And so they have slightly different predictions. I think that's an important distinction in the sense that, with the tRNA, that sequence basically can't change. It has to make the exact same function because it has to bind to a ton of different potential proteins or nascent mRNA molecules that are going to be proteins. And so there's slightly different predictions there, but the bigger idea of, do we need this redundancy, and do we need these certain protein coding genes in the same way that we probably need many, many tRNA genes? I'm not sure. It will depend on how frequently they're knocked out by somatic mutation if this model is correct. Obviously, other models may also predict this. And so that's part of the reason we're really excited to start getting into directly measuring somatic mutation rates across the genome and to start thinking really deeply about this. But I think it's certainly possible that the same sorts of processes are at play in other genes and potentially the ones that have real, functional important effects on development and biology.
All right. Well, thanks. It's to me, I feel that when we're starting to look at how evolution contributes to biodiversity, and then looking at it's one of the ways we might understand more about the functional roles of these species as they disappear. We don't actually get a chance to study them, but we still have their molecular information if we actually have the specimens. So it's sort of interesting from that perspective. Thanks a lot.
That's true. Yeah, it's going to be fun.
So I don't see any other hands up. Are there any questions that anybody would like to ask? Matt has a hand.
Who's that?
Hey, Russ. Nice talk. Really enjoyed it.
Cool.
I was interested in what you were talking about with the access to the germline with your introners. And it got me thinking a little bit about the mechanism. And I think you said something about horizontal gene transfer, but I was wondering how much we know about how those introners make it into the genome.
Yeah, the answer is not much. I mean, these were basically discovered in a conclusive way just a few years ago. So these are the early days. I'll also say that a lot of the organisms that seem to have introners are, I don't want to use the word weird, but they're less genetically characterized organisms than others. And so it's, some of these fungi are rusts that grow on a very specific plant. And so we don't know a lot about how they're really working molecularly. And so these are kind of some of the big questions I'm interested in getting at. To be clear, that's not proven. That's a hypothesis. So I think it may be related to this, but I don't know. We have to find out. Time will tell. But I think it's something we're pretty excited about. With respect to how they get in, I mean, transposons, like molecularly how they get in, transposons definitely transfer between species at pretty high rates. Not introners necessarily, but there's quite a few transposons that are way too similar in sequence between really divergent species that they almost certainly were transmitted horizontally at some time. So this happens, I guess is what I would say. Exactly how is an important question, but I have no idea. Someone else?
Thank you.
To echo other people and say thank you very much for the talk. It was a lot of fun. This is a question coming from someone who doesn't think about this stuff much at all, so I take it with a few grains of salt. But in the second part of your talk, in the intron section, you started off with the seeming paradox that introns are splitting up genes, and why might this happen? What might the benefit of that be? I might have missed something, but I didn't hear what the benefit of splitting up the genes might be. Was that something that I missed, or is that still an outstanding question?
Okay, that's great. I think I'm being a little too subtle when I set that up. I think the answer is it's probably not beneficial most of the time. And so there's been pretty interesting experiments done where people remove introns and nothing happens. That's not universally true. There's a few cases like the double sex gene that I showed where it really matters. So you absolutely got to splice that right. But the vast majority, I think, are probably just the remnants of processes like the one I'm describing here, where a transposon comes in. Over time, mutation causes it to be indistinguishable from any other intron in the genome, and it looks normal. It's an intron. But that's unproven at this point. It's just consistent.
So follow up to that, for the introner part of it, is there an advantage of having those inside genes rather than not?
Yeah, the fungi are cool, right? So there's a set of species there where something different is happening. We have some crazy ideas about them. I don't know for sure. One thing I don't talk about, but I can tell you, is that if you look at the stability of the folding structure of those introners in the fungi, in other words, I'm asking, how good are they at forming secondary structures? Do they have nice stem loops like tRNA genes, for example, where they can bind parts of themselves to each other by being reverse complements? In the fungi, they do that a lot. They're super stable sequences. So it may be that they have some role in shaping gene regulation when they insert, and it might be a way to rewire your genome at some rate. And so you see this quite a bit in the fungi, where these things are basically only in genes or they're super enriched for being in genes. And I tend to think that there is something going on there. And so we are working on trying to get RNA-seq data from a species that has an ongoing intron gain event. So there's individuals in the species that don't have some and some that do. And we're trying to just see, you know, does this modify gene expression levels or some other, you know, molecular phenotype that we can measure and understand. But I don't know yet. That's also kind of conjecture at this point.
All right, so I'm not seeing any more hands up.
Cool.
And so with that, as I've said at the end of all of our seminars, as of late, you know, we would now retire to wine and cheese and dinner and have a great time to probe you further and for you to find out more about, you know, what we can contribute to this talk. Just like we couldn't have our virtual breakfast this morning. Thanks so much for coming. It's been great to have you here and spend an entire day on Zoom with us. And we'll be in touch. We continue the questioning by email afterwards. So if you're willing to do that,
No problem. Yeah.
Reach out to you that way as well. So I'll give a round of thanks to Russ. Thanks so much. And we'll be in touch by email. So you take care.
All right. Thanks so much, guys.
You all.
Thanks, Russ.